Okay, welcome to our third installment for genetics and evolution. This is really like 10.2 and a half. We're going to just strictly focus on polygenic inheritance here. Last time we talked strictly about dihybrid crosses. So this is just sort of like building off of that, but kind of deserved its own lecture. So here we are. So let's talk about it. So polygenic inheritance is a single characteristic that's controlled by multiple genes. This gives rise to continuous variation. So what that means is continuous means that there's more than just like two options, right? Sometimes when we think of genetics, there's always like a dominant and a recessive trait, and that's it. In continuous variation, there's all shades, right, of specific colors. So like the two topics we're going to strictly focus on in this lecture is human skin color and wheat kernel color. So both of those, I'm sure that you've seen humans before, so you know that there's different skin colors. Maybe you haven't seen wheat kernels before, but there are different colors, so just like take my word for it on that one. So we're going to talk about how this single characteristic can be controlled by multiple genes. So skin color is the result of pigmentation, right? Melanin being produced in the skin. So skin color is thought to be produced by up to 4 genes. That is too much for us to deal with right now. So we're just going to look at 2 genes with 2 alleles each. So although this isn't like exactly how skin color works, the general idea of it is how skin color works. But if you thought like trihybrids were bad, now try doing it with 4 genes with multiple alleles each. So it's just like too much to put into a Punnett square and like show you. So we're just gonna pare it down for education purposes, 2 genes, 2 alleles each. So if we take these 2 genes, A and B, and they have 2 alleles each, a capital A will add melanin and a lowercase a won't, and same with the B's. Capital will add, lowercase will not. We're going to assume these genes aren't linked. And so again, in polygenics, alleles can be contributing or non-contributing. So the capitals are contributing and non-contributing ones are the lowercase b's. In the inheritance of skin color, when we're only looking at 2 genes with 2 alleles each, there are 9 genotypes possible. And you're probably looking at this picture over here and you're like, Ms. Kalfa, I can count, can you? Because like that's more than 9 genotypes. But it's not really, because this capital A, lowercase b is the same thing as this one. It's just in a different order. So it has all of the genotypes listed, but as you can see, some of them are repeats of other ones. So not really. It is 9 genotypes total. I guess I should say 9 unique genotypes total. And then you can see the 0 units means that these are all non-contributing. 1 unit is there's at least 1 capital letter. 2 units, 2 capitals, 3 capitals, and all 4 capitals. So when we're looking at this Punnett square, you can see that depending on what a parent's genotype is, the children's genotype can be lighter or darker than both parents. So when we are looking at this, the mother has 2 contributing alleles and the father has 3 contributing alleles. Well, when you make all of the gametes that are possible for this, for both mom and dad, and put them into this Punnett square, you can see that some of them are going to be darker and some of them are going to be lighter and some of them are going to be kind of in the middle, right? That's just how inheritance works, right? So then you can look at the F1 generation, which is at the bottom, right? And the phenotypes, right? So about, let's say 75% chance that the child is going to be a similar skin tone to the mom and dad, but it's definitely possible to be both lighter and darker. So let's talk about evolution of skin color, like global evolution. What are the advantages? So if you don't know this already, melanin has played a huge role in protecting skin from the sun. So and you can kind of see this in the map, in areas where it's right near the equator and it's much, much hotter and the sun is much more strong in those areas, you see a higher percentage of darker skin. And it's because it provides protection against UV rays. So skin damage and cancer, the melanin in the skin is protection. Whereas as you go further away from the equator, like up in northern Canada or like the Netherlands and Europe, you don't see as much melanin production because you need pale skin to get as much vitamin D as possible because there's limited sunlight. So you need to be able to absorb as much vitamin D as possible so that you survive, because you need vitamin D from the sun. Now we're going to talk a little bit, we're going to switch gears, we're going to talk a little bit about wheat kernel color. So we're going to go through this whole process of, we're going to cross a homozygous dominant for both genes with a heterozygous for both genes. And we're going to look at what the outcome would be. So here is my parental cross, right? Homozygous dominant crossed with heterozygotes. And here is my Punnett square. Now you'll note that I only did one row because, again, there's only one option for gametes for that homozygous dominant for both genes parent, so I don't need to repeat this 4 times. I just needed it once. And what that gives me is my phenotypic ratio. I have 1 very red because I have 4 contributing alleles, 2 red because I have 3 contributing alleles in both of the middle ones, and then 1 pink where I only have 2 contributing alleles, so it's like right in the middle. So again, let's just kind of cover this again. A trait is controlled by 2 genes, each with 2 alleles. How many genotypes and phenotypes are possible for this trait? Well, there's 16 combinations of alleles. So even though I'm getting 16 combinations, they're not all unique. So I'm actually only getting 9 unique genotypes. And within those 9 unique genotypes, a lot of them are the same. And so I'm actually only getting 5 different phenotypes. So I'm getting one that is like very red, right? All contributing alleles all the way on the left. And then I'm getting some red ones with 3 contributing, some pink ones with 2 contributing, some light pink ones with only 1 contributing, and then I have one that has no contributing alleles, which means that it would be white. So now, what if we were talking about 3 genes, 2 alleles each? So now we're talking trihybrid. So this is crazy on its own, and this isn't even, if we were talking about skin color for real, this isn't even 4 genes. This is only 3 genes. So now add in a whole nother gene and try and figure this out. It's just too much, which is why we pared it down to 2. But if I filled this out, and then I color-coded it, right, for contributing versus non-contributing, and you can see if you have capital letters, it is contributing lowercase. Non-contributing, you can kind of see the general flow from dark green to light green from the upper left corner to the bottom right corner, which gives me 7 phenotypes. And then if I were to figure out how many unique genotypes I have, I'm going to color code those as well. And I have 33 out of 64 unique genotypes. So you can see that it's a little bit hectic, and this is like kind of crazy. I would never ask you to do this on a test or anything, but it's good to see it and be able to actually understand how I'm getting these things. So again, you can go back and like go through all of this and see your unique genotypes and your unique phenotypes. So if I were to ask you questions about 3 genes, 2 alleles each, which I absolutely could, it would be more like this. So if you cross a homozygous dominant male with a homozygous recessive female, that's your cross, that's your genotype, and that's your Punnett square, because the only possible gametes are all lowercase versus all uppercase. And so your only possible offspring would be a heterozygous for all 3 genes. And chi-squared. You thought that was going away? That is absolutely back in action. So a chi-squared test, just to refresh your memory, is used to see if there's a difference between an observed result and an expected result. We use this statistical test to test for significance. The expected result is always going to be the predicted Mendelian ratio, and the null hypothesis is the prediction using Mendelian ratios, assuming they're not linked. So yeah, we're going to bring this back. We're going to do just a few practice problems. We spent a long time on it, so I'm not going to do anything crazy, but I just want you to be aware that chi-squared test is not just for ecology, it's definitely in genetics as well. All right, I'll see you for our 4th and final lecture next time.